Hello everyone, please go ahead and write your name in the chat box. In order to mark your attendance, please go ahead and do that. Okay, 16 people are there. Very good afternoon to all of you. I would request Tanuja to go ahead and talk to us. Other day you were not there for the orientation. Tanuja and Sumedha was not there. Tanuja, you're there? Can you hear me?
Yes, ma'am.
Okay, Tanuja. Would you let us see you? Can you turn on your camera?
Good afternoon, ma'am.
Hi. We can't see you, Tanuja. Okay, Tanuja.
Good afternoon, ma'am.
Hi, how are you? Where you been?
I'm fine, ma'am. I'm in my native place.
Okay, and what you did for 2-3 months? What?
I completed
Okay,
webinars,
okay,
courses,
hmm,
and, and work on my basics. That's all.
okay. And you are in your native place since?
Yes, ma'am.
So from 2-3 months you are there only at your native place.
No, 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 ma'am.
You recently went there.
Yes, ma'am.
Okay. So we are going to start with the online session from today. Today I'll be having just syllabus discussion and little bit of an overview of a unit which I'm going to start. Okay. So Tanuja,
Yes,
Sumedha
ma'am.
is there. We'll talk to Sumedha as we did not talk to Sumedha as well. Okay. You can turn off your camera.
Yes, ma'am. Okay.
Sumedha, you're there? I guess Sumedha couldn't join us. I hope everyone has written their name in the chat box. Please don't leave that because that's the only record I will be having with me in order to check whether you were there for the class or not. Okay. So please make sure that you are having your name entered in the chat box. And don't send it to me privately, send it to everyone. Even if you send it to me privately, but it becomes another record. So just send it to everyone. Okay. So 18 participants are there. I think few more are yet to join or they are not able to join or something like that. Let's see. But we are recording session. We should be able to upload it. I guess Ketan is not there. Okay, I'm sharing the screen with you people. One of you can unmute yourself. I'm unmuting Shabdali. Shabdali, can you hear me?
Yes, ma'am.
Shabdali?
Yes, ma'am.
Dhanashree, you're there? Can you guys hear
Yes,
me?
ma'am.
Okay.
Yes, ma'am.
Yes,
Yes.
ma'am.
Okay. So I'm sharing my screen. Just let me know when you guys are able to see it. Okay. But now just stay unmuted for a while. Can you guys see the screen?
Yes, ma'am.
Yes, ma'am.
Yes,
Yes.
ma'am.
Okay. Okay. Since I'm thinking that you guys are some people are in the native place, some people are at home, and I don't know. They must be having their books and syllabus with themselves. But let's go over. I do not know the exact pattern of examination and stuff like that as of now, but we'll be regularizing it soon as we get the guidelines from UGC. Okay, and this is your semester 3, paper 1 301, paper 2 302, paper 3 303, paper 4 304. So we have in 301 is regulatory microbiology, as the basics of microbiology were done in semester 1 and 2. Then you have genomics, then you have toxicology and also nutraceuticals. Okay, and practicals with regards to that. So as of now, we are not sure what are we going to do about the practicals, but we can always go ahead and learn the theory. Then this paper 2 belongs to MS application, so mass spectroscopy in detail, starting from this semester, which will be extended to the next semester. So you'll be having MS applications, LCMS, GCMS, etc. Then you'll be having metabolite studies, thermal analysis, and tracer techniques. Then in paper 3, you'll be having standardization of ASU. Now you know that what are ASU, what are ASU products, etc. And if you have a ASU product, how will you go ahead and standardize ASU drug? So that is what it is. Then you have statistics and GMP. Statistics would involve biostatistics and good manufacturing practices. Okay. Then you have BAB studies, GCP, and method validation. Okay. BAB studies is nothing but bioavailability and bioequivalence studies, good clinical practices, and method validation. Okay. So these are the things that you have. You guys have 4 papers, 4 theory subjects. Each paper will be having 4 units. As of now, you guys have, know that very clearly. And we'll be learning each unit for 15 lectures. And we'll see how it goes about. Since it's going to be an online session, it might take a little longer, or sometimes we can also go ahead and finish it up little quickly. Okay. So for me, I'll be starting with paper number 4, which is PGBS 304. It involves BAB studies, introduction to good clinical practices, and method validation. Okay. So let's go ahead and check what exactly this paper holds. Okay, so we are here in semester 3. Let's go to paper 4. So paper 4, I guess you guys can see it properly. Can you guys see it properly? Somebody can unmute and tell me.
Yes, ma'am. Yes, ma'am.
Okay, so first we have is ethical issues in clinical trials, and what all we'll be learning. Oh, these are the learning objectives. So let's go to unit. This is just a minute. Sorry for that. Ethical issues in clinical trials, so origin of ethical issues, dealing with ethical issues, ensuring the compliance to ethical issues, what is ethical committee and their setup, then regulatory powers of ethical committees, and ethical issues in animal studies, how do you compliant with this, how do you become compliant with these ethical guidelines, then dealing with ethical issues, etc. are going to be covered in this ethical issues in clinical trial. Then you have GCP. GCP will be again talking about when did the origin of GCP happen after going through all the ethical issues. Then we are going to learn about the guidelines of GCP with respect to ICH and ICMR. This is sorry over here. So this is ICH and ICMR. Okay. So the next unit will be bioavailability and bioequivalence. So it's nothing but you'll be learning a simple introduction to bioavailability and bioequivalence, and what are the factors that affect the bioavailability and bioequivalence. What are the parameters that will help you evaluate bioavailability and bioequivalence. And then you have is analytical method validation, which will include, so whenever you have an analytical method, या फिर protocol concerned with a machine, with the instrument, analytical instrument, how will you go ahead and validate that particular instrument along with that protocol? So that is what this unit is going to going to cover. So for me, I'll be starting with ethical issues in clinical trials. So now we know how important are the clinical trials, as we are all facing this pandemic, and we know all of us are waiting for the vaccine to come into the market. But there are so many hurdles, there are so many things that a researcher has to take care of. that before starting a clinical trial. And ethical issues becomes a part of the clinical trial. So ethical issues, this chapter is basically named as ethical issues because in that era, when people were trying to search new medicines and they were trying to do some research with regards to this medication, they did not know how would they go ahead and evaluate what is the exact effect of that medicine on person's body. So in that case, they would just enroll people without their knowledge, without taking any consent from them. So in that case, many people died because of abuse of medical products without their consent. Because see, for a researcher also, it was like a trial error कि ये medicine काम करेगा या नहीं करेगा उनको पता नहीं. But then they went ahead and still tried on people without knowing the after effects and without giving them any exact compensation. And also they did not let them know that this is a new kind of a product that we are going to try on you. And in that scenario, many people died. So in World War II, I guess, yeah, in Hitler's era, he enrolled many prisoners and Jewish people for clinical trials without their consent. And in that case, what happened was many people died because medicine, we all know that every medicine has a side effect. And especially when it's a trial and error medication, you do not know what exactly that medication is going to do to you, right? And then we also know about thalidomide this thing side effect. So thalidomide was a drug that was given for the morning sickness for pregnant women, and they did not know about the means they did not went ahead and do any kind of what you say clinical trial या फिर side effect studies for that particular drug. And what happened was that drug was acting very good for the morning sickness, but it had a side effect on the baby who is there in the womb. So all the women who took thalidomide as a drug for the morning sickness, they had the babies without limbs. So that was the big disaster in the world of FDA at that time. And then that is when the rules started becoming stringent. Rules started becoming stringent in the sense that you have to prove that your drug is there, it's a good drug, and it has no side effects. And which form of the drug, that is D form, L form, you guys must be knowing since you guys have studied a little bit of a chemistry. It's a basic chemistry. You have every chemical formula has a D structure and L structure. So many a times these are like two mirror images of each other. So even if you change the D form to L form, uh, being taking into consideration that the chemical formula is the same, but it might turn into a altogether new drug and show some other side effects on the body which are not supposed to be there. Okay, so this is what people are trying to tell, means like this is what this unit is trying to tell you, like what all happened in the history and how and slowly and steadily we started developing some rules and regulation in order to conduct the clinical trials. Right now also when we studied in Sem II, we studied I think new chemical entity. In that also we talked about what is the flow of a drug discovery and how much time the drug discovery takes, right? So you start with 10 molecules, out of which one molecule will be selected. From that one molecule, you will go ahead and do the animal toxicity. That means you will do the animal studies in vitro, in vivo, and then you will go ahead and do the clinical studies. So you imagine in that case, and as we studied in the last semester, it takes around 10 to 15 years for a drug to come into the market. And now you imagine we all are facing this pandemic. This whole world is facing the pandemic. And what you think should be happening right now in order to, in terms of searching a new vaccine for this coronavirus? So it is a very biggest challenge for all the researchers who are working on this line and really working very hard in order to find that one molecule. You imagine that finding that one molecule we had talked about, it took around 2 to 3 years. And currently what we are expecting, all the people who are sitting back home, people like us, we are expecting that that molecule should be searched within 1 to 2 days, 1 to 2 months, right? Because nobody wants to stay at home anymore. Everybody wants to go back to their normal life. But we all know that there is no solution to it until and unless there is a vaccine into the market. Because if you do not take care, you are going to face you are going to face the coronavirus. People might, the people like us, like who are young, I'm not very young as that of you guys, but we are young in that particular category, where we might not get any symptoms, or we might not get affected at a very high rate as compared to the people who are in the critical situation, or comes into that or come into that periphery. But end of the day, you might get it or you might not get it. Unless and until you don't have a vaccine, you just can't do anything about it. You cannot roam around freely, you cannot do anything. And so hopefully people will develop some kind of a immunity, or some herd immunity, that concept you are thinking people are learning about right now. But that will take a long time. For that, 40% of your population has to be affected by coronavirus, and 40% of 1 million people is going to be very, very, very huge number. So that is what it is. So if you try to think about this, taking this pandemic into consideration, and thinking about it, what could be the clinical issues that researchers must be facing? Can any of your, and like people from you guys can tell me any issues that researcher might be facing? One by one, you can unmute yourself without making any noise. If you think of something that what researcher must be going through right now, you can tell me. I give you 2 minutes to think about it. Am I audible?
Yes, ma'am.
Okay. So you want to go ahead and tell me what researchers must be facing, what kind of a issue they must be facing right now in order to develop, अभी वैक्सीन डेवलप करने के लिए उनको क्या तकलीफ होती रहेगी, what they must be thinking.
Maybe the target molecule they are facing difficulties, or
yes, target
like,
molecule is a very, that's the most appropriate answer, that finding that target molecule is the most important part, right? Because without the target molecule, you do not know what exactly you are which area you are going to target, which part of the virus you are going to target exactly, right? So target molecule becomes the most important part. So that could be one of the issues that researchers must be facing, because for that you have to study that virus in detail, with genetics, with the structure, and etc. Because when you want that virus to be killed, you want means like if you want that virus to be killed, or your immunity to be developed against that virus, you have to know core structure of that virus, and also the genetics of that virus, so that you can go ahead and make a molecule in such a way that will help take care of the virus in such a way that our body will develop immunity for the same, right? And currently medicines are also doing the same. Medicines are targeting the viruses in different fashion, so that virus will be killed and body can recover, right? All the antiviral drugs that Glenmark and Cipla are providing, right? And what else? What next issue? In terms of clinical trials, we are talking. So first was the told by Musrat was this thing, target molecule difficulty. Yes. Next one. People can go ahead and tell me.
Ma'am,
Hmm.
maybe the ensuring the safety of the vaccine or drug, that it has less side effects or it can't harm the patients.
Yeah. So ensuring the safety of the vaccine. So, but if once you have to try it on the subject, but before that you will be doing the preclinical trials, right? And the preclinical trials would involve doing the same vaccine testing on the animals. So currently, if you think about that, finding the target molecule and next moving ahead and checking whether this vaccine is going to work or not, first you have to try it on the animals. And also while trying on the animals, you will be checking that whether this vaccine is working and working or not. And secondly, you will be checking that what kind of a toxicity it has on the animals. So first you will be targeting the animals. So that could be one part. So उनके कितने तो batches ऐसे रहेंगे animals के जो work ही नहीं हुए, right? And कुछ some of the animals or some of the trial molecules did not work on the animals. So if you screen 10 or 12 target molecules, you will go ahead with one or two, right? So चलो हमने मान भी लिया अगर एक target molecule ये हो गया है जिसने एनिमल टॉक्सिसिटी भी पास कर लिया है, व्हाट वुड बी द नेक्स्ट इशू?
Designing of drug.
One person designing of the drug, and other answer I could not hear. Hello.
Hello.
Yes, Musrat, you told me that designing of the drug, and somebody else, some male voice told some answer. I don't know who was it. Akash, was it you?
A mode of administration.
See, drug designing will be taken care at the time of drug discovery itself, right? Because you cannot even try the medication on animals unless and until you design a route of administration, right? So when you do the in vitro studies, maybe, and in the lab, you can use the drug in the liquid form or something like that. But when you are going to proceed through animal studies, you will have to think about the route of administration. And route of administration plays a major role. ये भी हमने last 2 semester में देखा है कि route of administration also plays a major role in order to know that how much of the drug will be bioavailable, how much of the drug या फिर vaccine in this situation will be bioavailable. Now you guys must be reading the news. People are coming up with the nasal vaccine, people are coming with the injections, people are coming with the oral ones. So there are many ways of it. Okay, na? And the one way you want to think about is like which will make sure that it is available in the blood as soon as possible, so that people will go ahead and develop the immunity for that particular virus. So drug designing was one part that people might face difficulty while detecting. So that comes into drug discovery and drug designing, and then you will be thinking about the toxicity of this particular vaccine on animals and effect of that particular vaccine on the animals. ठीक है, इसके आगे क्या होगा? इसके आगे ये होगा कि which people you will select for the clinical trial, because you will not be selecting sick people over here, because people who are already have coronavirus, will you go ahead and select those people? No, because you are doing the vaccine trial, you are not doing the drug trial. Drug trial you might do on the people who are sick already due to COVID-19, but when you're trying to develop a vaccine, you'll have to select a population who is healthy enough. Okay, So people must be also, researcher must be also facing a problem with selection of this healthy subject in this particular pandemic era. And also how many, mic is, yeah, somebody's mic was unmuted. So yeah, and what kind of people you will decide, you will take in your trial? Obviously, people will need healthy population to go ahead and enroll into this clinical trial. And imagine, see, for normal drug trial, it is very easy for people, healthy subjects, to go ahead and enroll for any kind of a clinical trial. So generally कैसा होता है, जब भी clinical trial होता है, people are given consent, means like उनको एक पेपर दिया जाता है जिसमें in detail सब explain किया हुआ रहता है कि this is what is clinical trial is going to be about, this is our drug, this could be the possible side effects. We are also we have made the same drug as that of another company which is available in the market or something like that. So this is what it is. Would you like to go ahead and enroll for the clinical trial? So people will go ahead and enroll for the clinical trial. But in this condition, imagine in this pandemic condition, people are already scared. People do not want to even leave their house and go outside world and see what exactly it is. Maybe 20 to 30% people do not think in that way. But I'm sure 50% and above people are thinking that you should be safe at home. People should not take risks, or we should not go ahead and take risk and jeopardize our lives and our families' lives. In that case, it becomes very difficult and challenging for researchers to find healthy human beings who would go ahead and enroll into this kind of trials. Okay, so this could be, and in case of ethical issues also, in let's come to this. So this was we talking about COVID-19 and what all researchers must be thinking at this point of time in order to develop a drug molecule, then design the drug molecule, then go ahead with the preclinical trials, and then go ahead and develop a go ahead and enroll into the clinical trials. और clinical trials के भी 4 phases रहते हैं, ये हमने देखा है, phase 1, phase 2, phase 3, phase 4. So phase 1 से शुरू होके, pilot study से शुरू होके, phase 1, phase 2, phase 3, and phase 4 is nothing but marketing. So it's going to be so, I don't know what is going to happen once the vaccine comes into the market, whether we will, common people like us will be able to get it or not, or how much of the money we'll have to pay for this, because everyone is so eagerly waiting for this virus. So let's just hang in into the situation and see how exactly things are going to happen once the vaccines get developed. So coming back to this particular unit, which now we know that these 2 units, 304.1 and 304.2, are going to be related to your GCP, means like good clinical practices. And you will learn now like what researchers and what companies, what kind of a protocol they are following when they are conducting the clinical trials. Okay? And in this particular unit, for slides will be starting from tomorrow. So in this particular unit of ethical issues in clinical trials, we are going to see that what are ethical issues, जो human right है, that I am a human, I have all right to live my life the way I want. I do not want anybody to rule me without my consent. या फिर I don't want to do anything because other person is telling me to do without and making me do without my consent. So ethical issues in clinical trial, how did they originated, what all incidences happened, and what all things were taken care of. Like you will be learning about Belmont report, you will be learning about Nuremberg code, and you will be learning about many more things ahead, which are very important in terms of developing this guidelines for good clinical practices. That is what is we are going to see in this particular unit. So we have hardly 3 minutes left for this 40 minutes lecture to get over. So what I'll do is I'll go ahead and stop over here, And tomorrow we'll meet at around same time, either 11:30 or 12 o'clock, and start with the slides for ethical issues in clinical trials. I hope you guys have understood a pre-discussion about this particular unit and what exactly clinical trials are, how exactly clinical trials must be taking place in for developing this particular vaccine in this COVID-19 situation. And as and when we proceed ahead with this particular unit, today I'm going to create the Google Classroom for you people. I'll be posting the assignments which you guys are going to write and submit on daily basis, या फिर 2 days में एक बार, ठीक है? So thank you very much for being there. I hope everyone has written their name in the chat box. 21 participants are there. I hope the 21 names are going to be there. So make sure you write your name always in the chat box. And tomorrow we'll meet again. Okay. Bye everyone.
Bye, ma'am.
Bye.
Bye.